Okay, so now we're going to move on and talk about the dihybrid cross. Now, a dihybrid cross involves two pairs of contrasting traits. So once Mendel got all his data from monohybrid crosses, then he decided to start studying two traits at a time, or two characters at a time. So round seeds and wrinkled versus round and wrinkled seeds versus green and yellow seeds. So both seed shape and seed color. Again, the parentals are all going to be true breeding for each individual trait, so he's going to have all plants that have smooth seeds and versus plants that have wrinkled seeds, and plants that have green peas versus plants that have yellow peas, and they're going to be true breeding for each trait. Now, when he did the crosses, when he did the parental cross, again, you end up with an F1 generation that is uniform, all the plants look the same, and they're all carrying those dominant phenotypes. And then in the F2 generation, both of the recessive traits show back up again, and in a very predictable way that led Mendel to form his postulate of independent assortment, which we'll be talking about shortly. So when you look at this cross, you can see now notice these are two different crosses. You have yellow and round, which are two dominant traits, and green and wrinkled, which are two recessive traits. And when you cross those two, you get all yellow round. But if you mix it up a little bit and you take a dominant color and the recessive shape in one parent, and the recessive color and the dominant shape in the other plant, you end up with the same results here. So it doesn't matter if the parent is dominant for both traits or not. You can mix it up and you end up with the same results. They're all yellow round. They're all showing those dominant traits for both characters. Then when he performed the F1 cross, he ended up with a ratio of 9 to 3 to 3 to 1, where a majority of the plants still show that dominant phenotype for both traits, for both characters. So a majority, 9 out of 16 of the peas are going to be yellow and round. And then 3 out of 16 are going to have one dominant and one recessive trait, so yellow and wrinkled or green and round. And then only 1 out of every 16 plants is going to have that both recessive traits, so green and wrinkled. So like I said, note that the parental cross yields all yellow round. And it doesn't matter if the dominant traits came from the same parent or not. And then the F1 cross results in that 9 to 3 to 3 to 1 ratio. So looking at that, looking at the Punnett square, you can see that it doesn't matter for, so like here for parent 1, we've got the parents are yellow round or green and wrinkled. So these are the possible gametes that can get produced, and then this is the resulting, here this is the resulting F1. If you switch it up and you have the parents have one dominant and one recessive, but they're still true breeding, and then the second parent is the opposite, you end up with the same result here. So that's what I was saying. It doesn't matter if each parent is like all dominant for both traits and the other parent's all recessive for both traits, or it gets mixed up. The results in the F1 cross will be the same. Now, the reason that this works the way it does is because of independent assortment. And so we have the luxury of hindsight, and we can say, oh, well, that makes sense because we know how cells divide and we know how meiosis works. We just went through all that in chapter 2. But Mendel didn't know anything about that. So the fact that he figured this out without that knowledge is really quite remarkable. But we know that the reason it works is because these unit factors, what he called unit factors, we now know are genes on chromosomes, get that assort independently of one another, and that you can have all possible combinations of gametes formed during the process of meiosis. Now, an easy way to figure this out is, when you've got a dihybrid cross that you need to do and you want to know what all the 4 possible gametes are, and we actually did this as your homework for chapter 2. An easy way to do that is just for each letter, so the first letter here, big G, you want to look at the 2 ways that it can combine with the other alleles. So you've got big G, big W here, and then big G, little w here, and then little g, big W here, and then finally, lastly, little g, little w here. And so that's just a kind of a quick and easy way to come up with the 4 possible gametes that get formed during meiosis for each parent. So then when you set up the Punnett square, again, you just line up the 4 possible gametes for each parent along the side here and along the side on the top here. And then you start combining them and you look at all the different combinations. And you can see here, when you add up you get a lot more going on with the genotypic ratios, but when you add them all up as far as phenotypic ratios, you get 9 to 3 to 3 to 1 for the 4 different possible phenotypes. Okay, so Mendel's 4th postulate is independent assortment, and it states that during gamete formation, the segregating pairs of unit factors assort independently of one another. So this means if you have, like we've been talking about, the smooth seed and wrinkled seeds versus the yellow and green, like you can get all possible mix-ups. It's not that green always goes with wrinkled and smooth always goes with yellow. They completely assort independently of one another, and we know that that's because they're on different chromosomes. So really what a dihybrid cross is just 2 independent monohybrid crosses, and the 2 contrasting traits are inherited independently one another. So you can really look at it as just independent monohybrid crosses, and you don't have to do a big dihybrid cross with 16 different squares to fill in. You can just do 2 monohybrid crosses and then just multiply the probabilities, because we know that the product law states that when 2 independent events occur simultaneously, the probability of the 2 outcomes occurring in combination is equal to the product of their individual probabilities. So you just multiply them. So you can see here, so this is if we were just looking at yellow versus green here. And we know that the results of a monohybrid cross is 3 to 1. So 3/4 are yellow and 3/4 are green. Then this is our wrinkled and round monohybrid cross. And so here we have 3/4 round, 1/4 are wrinkled. And then you have to do it a second time because we want to compare against all the different possible combinations. So that's our first monohybrid cross. This is our second monohybrid cross. Now we just multiply across. So you notice here that you've got 3 times 3 is 9, and then 3 times 1 is 3, and then 1 times 3 is 3, and 1 times 1 is 1. So that's where you get that 9 to 3 to 3 to 1 ratio just by multiplying across. And this is called a branch diagram. And I think it's a lot easier to deal with than a Punnett square, personally. But you just kind of have to get used to it. But you just have to recognize that each individual cross is just a simple monohybrid cross, and then you just got to remember to multiply them through to get your probabilities. Okay, this is another animation from the book that just basically reviews everything we've already been talking about. So, in a dihybrid cross, you're looking at 2 different characters simultaneously, when we do the cross, we know these are all the possible combinations that we could get of phenotypes. So in the parental cross, when we cross the yellow round and the green wrinkled, we get all yellow round. So all the F1 progeny are show that dominant phenotype. And then when we do the F2 cross, we get all the 4 of the possible combinations with a very predictable ratio of 9 to 3 to 3 to 1. So, you know, looking at those laws of probability, we can look and figure out how we can multiply them out. So individually here we've got yellow or green is going to be a ratio of 3 to 1, and round wrinkled is going to be a ratio of 3 to 1. And if we look at all the possible combinations of these words, so yellow, round, yellow, wrinkled, green, round, green, wrinkled. Those are our 4 possible combinations of the phenotypes. And so now all you have to do is multiply the probabilities. So here we've got yellow is 3/4, round is 3/4, there gives you 9. Yellow is 3/4, wrinkled is 1, there's your 3. Round is 3, green is 1, there's your other 3. Green is 1, wrinkled is 1, there's your 1. So that's where we get our 9 to 3 to 3 to 1 ratio. So you can also do a test cross with dihybrid, so where you're looking at 2 characters simultaneously. And so you've got, this time you've got a plant that is dominant for both traits, and you want to know if it's heterozygous or homozygous. Again, you just cross it with a homozygous recessive. And we know that if they were all here, if they were all homozygous, then you would get, you know, all the progeny would be dominant, because that would be the same as a parental cross. So here we're going to look at it as if they were both heterozygous for the trait. So the possible gametes that can get formed, over here we've got all 4 of those possible gametes. Over here there's only, you know, you can write it out 4 times for your Punnett square, but there's really only one type of gamete that's going to get produced, and that's that because there's only recessive alleles. So it's all going to be lowercase g, lowercase w. So when we create our Punnett square, we put, you know, one parent, set of parents along the side, and another set of parents along the top. And then when you start combining them, you can see you end up with 4 to 4 to 4 to 4, or 1 to 1 to 1 to 1. So our phenotypes are all going to show up in equal amounts in a test cross with a 1 to 1 to 1 to 1 phenotypic ratio. Now, to make it even more complicated, you can do a trihybrid cross. So if you look at all the possible gametes that can be formed in the F1 for 3 traits, 1, 2, 3, you end up with 8 possible gametes. Now, all a trihybrid cross really does is just show even more that all of Mendel's principles hold true. When you do that F1 cross, you get 8 possible gametes. So if you were going to do a Punnett square, you'd have to have 64 squares to fill in. And since we know that it's really just 3 monohybrid crosses all put together, and we can use the product rule, it's much easier to do a branch diagram for a trihybrid cross. And so here's a branch diagram here, and you can see we've got, this is your first monohybrid cross, this is your second monohybrid cross, and this is your third monohybrid cross. But you've got, when you do a branch, you've got to create new branches in each one so that you can compare them all. And then you just multiply across. So here you've got 3 times 3 times 3, which gives us 27. 3 times 1, or 3 times 3 times 1 gives us 9. Then 3 times 1 times 3 gives us 9. Then 3 times 1 times 1 gives us 3, like that. So you just keep multiplying across. 1 times 3 times 3 gives us 9, and you end up with a 27 to 9 to 9 to 9 to 3 to 3 to 1 ratio. So you have 8 different phenotypes, and 27 different genotypes. Now it would be difficult to look at all the genotypes on a branch diagram, but once you get to tri-hybrid crosses, we're really not concerned about all the different possible genotypes. We're more interested in the phenotypes at that point. So to make things even a little bit more simplified, there's a little shortcut trick that you can do. So notice on the previous slide it said that there was 8 genotypes for a tri-hybrid cross. We know there's 4 different genotypes for a dihybrid cross. So if you look, you can, or gametes, not genotypes, gametes. So if you look, if the number of heterozygous gene pairs here, so this would be monohybrid, dihybrid, trihybrid, like that. If you multiply or use 2 to that number, you can determine the number of gametes. So if you have a monohybrid cross, then you're going to have 2 gametes. If you have a dihybrid cross, you're going to have 4 gametes. If you have a trihybrid cross, you're going to have 8 gametes. And then the genotypes produced are 3 to that number. So 3 genotypes, remember that 1 to 2 to 1 ratio in the monohybrid cross. There's 9 different genotypes that could get produced in a dihybrid cross, and we said there were 27 in a trihybrid cross. And then the number of phenotypes produced is the same as the number of gametes. So you have 2 being produced in a monohybrid cross, 4 being produced in a dihybrid cross, and 8 being produced in a trihybrid cross. But what's really important to note is that's only if the organisms are heterozygous. These calculations don't work. You don't get the same ratios when you're crossing, doing a parental cross or a test cross or something like that. So this is only for F1 crosses when the organisms that are being crossed are heterozygous for their traits. So when Mendel was doing his research, it was around the same time that Darwin was working on his origin of species, and he was looking at evolution. And you know, the idea that how evolution would work would be something rather continuous, like parents produce an offspring that's kind of a blend of both parents and kind of get like the best of both worlds, I guess, and so then that gives them an advantage, evolutionarily speaking. That's kind of what the thought process was at the time, where Mendel has this discontinuous variation, that these, you know, recessive alleles pop up. They're masked in one generation and they pop up in the next generation, and that didn't really jibe with what the thinking was of that time, and so people pretty much just ignored his findings. And it wasn't until the early 20th century when we started looking at chromosomes and we actually identified chromosomes. Walter Fleming discovered these chromosomes, and they started doing experiments with them, and they started seeing how these chromosomes do assort, you know, what happens during meiosis. And they were doing literary searches, and they stumbled on Mendel's work, and they recognized, wow, what Mendel saw really goes along with what we're seeing is happening in these cells when they divide, and it, you know, it meshed very well with the chromosomal theory of inheritance, that genetic material is contained on the chromosomes. And so these unit factors that Mendel was talking about how they segregate and how they assort independently, you know, that's what they were seeing in a microscope with these chromosomes. And so it's kind of all came together, and it all made sense. So just kind of a recap of everything we've been talking about, both from Chapter 2 and Chapter 3 now. We know that each species possesses a specific number of chromosomes in their somatic cells, and that number is considered the diploid number, or 2N, and so that consists of 2 chromosomes or 2 homologs carrying 2 alleles, or 1 allele each, so 2 alleles total for every trait that we have. And during gamete formation, that diploid number is reduced by half. so that we produce these haploid gametes that then combine through sexual reproduction to produce the diploid cell again. And we know, again, that this diploid number is comprised of these homologous pairs, and that each gamete contains one member of each pair, and that the number of chromosomes in the gamete is half of what's in a diploid cell, and it's called the haploid number. And we know again that genes are the unit factors that Mendel was describing, and that they're located on these homologous chromosomes, and that members of these homologs separate or segregate during gamete formation. So like I said, all that stuff kind of all came together. The researchers looking at the chromosomes and looking at cell division put together what they had found with what Mendel had found, and then it was just like, you know, everything made sense. Now, with Mendel's principle of segregation, we've already looked at these pictures before, so just kind of a review, that Mendel had these paired unit factors, which we know are actually alleles present, you know, alternate forms of genes present on homologous chromosomes, and that they pair up during meiosis and then get segregated independently of one another. And then you have all the different possible combinations of gametes that can be produced through that independent assortment. And that's as long as these genes here are on different chromosomes. Note they're on different chromosomes. We'll see later in the semester that if the genes are present on the same chromosome, these laws don't necessarily apply. And so that's why it's really good that all the 7 characters that Mendel looked at, they were all on separate chromosomes. Again, he would have gotten really confused if that hadn't have been the case. So one of the great things about independent assortment, and this is something that we talked about in Chapter 2, is that it leads to such extensive genetic variation. So if we look, you know, 2 members of any homologous pair are rarely genetically identical. You know, you're going to have, you know, when a mom and a dad decide to have offspring, of course their chromosomes are going to be very different. So when in that individual offspring, when their gametes are formed, you know, those gametes are going to be very different from one another. And during gamete formation, all possible combinations of maternal and paternal homologs are produced. So if you look at some of the math, some of the probability we've already talked about, we said that the possible number of gametes produced is 2 to the n. So if we're looking at humans, because we have 23 chromosomes, right, in our haploid number, then that means that there are over 8 million possible gametes that are produced just by one individual through independent assortment. That's not even really talking about crossing over and all that. And then fertilization. So if you take dad's 8 million different combinations and combine it with the mom's 8 million different combinations, you get over 1 trillion combinations that can occur. So what that means is if 2 people were really busy and they tried to have 1 trillion children, that theoretically speaking, they could have 1 trillion different children. They could not have the same child twice. So when, you know, when they always say you're 1 in a million, it's really more accurate to say that you're 1 in 1 trillion, and that every person on the planet is unique. And this is really important for the process of evolution. All right, so this is it for the second lecture. So be sure to answer all the review questions that are associated with this lecture, and then you can move on to lecture 3, where we are going to start talking about some math and doing some probabilities. All right.